chairperson of the Programs Committee, and she will introduce our guest speaker for tonight. Catherine?
Thank you, Dr. Roach. Welcome, everybody. It's wonderful to see everybody together again, friends, and I'm sure lots of relatives in the room. I'm excited, as you are, to hear about the results of the DNA study. I want to introduce our speaker at this time. We welcome a good friend, Dr. Raquel Fleskes. Those of you who participated in the Chesapeake Research Project back in 2019 will recall that Raquel, along with her then PhD advisor, Dr. Theodore Scher, interviewed each of you and collected your DNA samples at Tudor Hall. In February 2020, just before COVID hit, she and Dr. Scher presented their findings to a large audience at the College of Southern Maryland. Well, Raquel is now Dr. Fleskes and is with us again to present her research findings, and we are confident that you will find her presentation engrossing. As we prepared for these remarks to introduce our speaker, we were struck by, well, the depth and brilliance that Dr. Fleskes brings to her role as researcher, teacher, and mentor. Earning her PhD in anthropology last year from the University of Pennsylvania, she won a prestigious National Science Foundation postdoc fellowship at the University of Connecticut. On her resume, we counted no fewer than 37 peer-reviewed papers, conference presentations, and articles that she has authored. Her research has been covered extensively by the media, including publications ranging from National Geographic to Forbes magazine to the Washington Post. To list her affiliations, honors, and awards would extend this brief introduction into a lecture itself. Instead, we will simply end by saying, ladies and gentlemen, please welcome Dr. Raquel Fleskes.
Good evening, everyone. Good evening. Thank you so much for having me here today. It is really an honor, and thank you for that nice introduction from Dr. Bose and for Peter Laporte for inviting me here today. It's been about, 3 years since I've been back here. As Dr. Bose says, we gave a preliminary result, a presentation on the preliminary results back in 2019, and little did we know that was about 3 weeks before everything shut down from COVID. So we kind of had to slow the project down, just kind of how life a little bit slowed down. So when Peter reached out to me, I was like, this is going to be great because, we have to, we are here today, kind of restart life, right? Restart research projects. And so I'm really I'm excited to be here to kind of share with you where we are right now. Now, I'm going to preface this by saying this project is not done. Like many research projects, if you guys are all genealogists here, there's still many stories and things still yet to discover. So there will be no crazy relativatory insights, unfortunately, today, but rather kind of a visit as to what we know, how we know it, and then to share with you guys a little bit about some other research projects that are encapsulated in this Chesapeake DNA project. Okay, so now I'm going to try to navigate. All right, and this is the introduction slide. All right, so how do we know about the past? We're all here today at a historical society because we're interested in understanding where we come from, the history of the place that we live, the different times, the different stories, right? And oftentimes we'll use things like archival sources, oral history. If we have genealogists in the room, we're really into kind of reading those primary source documents to understand the stories of the peoples that make our family, right? And if we think about it at a local level, the stories that make, let's say, St. Mary's, this town, its town. Now, in addition, not all sources, those survive, right? So using, again, that genealogy, not all histories are recorded. Sometimes if we go back, we may not be able to, we may trace one line really well, and then other lines you might just get a dead end, right? And that could be because of things like class, race, sex, age, all these things. So we can try to use other approaches to understand the past. This can include archaeology, And methods in science, archaeology, looking at the study of material culture, the artifacts that get left behind, right? Or the foundations. You could think of maybe even Tudor Hall as being an archaeological project, right? There are many, many people that have come to live in Tudor Hall. Now, what I do is I look at the past using scientific methods. So this can include things like radiocarbon dating, isotopic analysis, or these are the cool geochemical signatures that go into our bones, but also DNA. They all provide windows into understanding our past. Now, what I try to do is combine all these different ways of knowing, all these different doors into looking into the past, and not to privilege one viewpoint or another, right? Archival work really provides a very deep foundation for the questions that we want to ask. Archaeology provides evidence for people who have come before us, and DNA provides kind of a stepping stone when we don't have documents and we may not have archaeology. So this is where the Chesapeake Ancestry Project has kind of tried to occupy that space in between, that liminal space. Now, why the Chesapeake? We could do this at any other kind of context, right? But you know what? I think the Chesapeake is a really important place and a really important time. Chesapeake, I grew up in Maryland locally, hearing old stories of families that live in this area that have been here for hundreds of years. And in conversations with you guys today, I continue to hear these stories, right? People, well, I was on the Ark and Dove, or, my family was a Fenwick, and I've been here, and there's a street that's named after me, right? So you have these really old stories, and it just got me thinking, well, if we have these people that have been living here for hundreds of years, how does that impact our DNA, right? How does that impact how we understand our history? Now, I'm considering the Chesapeake today as being both Maryland, Virginia, and present-day Delaware. Now, if anybody, you guys are big Delaware historians here, you'll know that Delaware is a little bit of a persnickety place. It was, in the 17th century, claimed by the colony of Maryland. But then later on, also, the colony of Pennsylvania and New York also tried to claim Delaware. But because we're thinking about the 17th century, and Maryland was really trying to get their claws on Delaware on this peninsula, we're going to consider it part of the Chesapeake region, okay? And we know the Chesapeake was also the first region to be permanently settled by British colonists. We're thinking about Jamestown, the 1607, right? And Jamestown was really just the start for a larger migration of European immigrants, right? And has a very unique demographic history. By 1700, there is thought to be approximately 100,000 English migrants living in the Chesapeake region. Now, likely there were many, many more individuals that came over, and we'll talk about that in just a second, but we have a very high death rate. So folks were coming from all across England. This is a map from James Horn that shows where folks that are indentured servants are coming from in England. So this is a map of England, right? So you can really see they're migrating from across the country. And much of what we know about the Chesapeake region and the migration comes from understandings of indentured servitude, or folks that would, in order to pay for their transport across the Atlantic, would basically sign a contract for a number of years of labor to pay for that, right? And we have really great records about who those people were, how many they came over, and what the demographics are, right? So going back to that indentured servitude, right, that lens of we have about 100,000 migrants living here by the end of 1700s, but probably a lot more that came over. Well, and that's because the Chesapeake is known for a very notoriously high death rate. It was not a really great place to be in terms of the 1600s. Sometimes in the 1640s, it was as high as 2 out of 3 indentured servants that are coming over here are thought to have passed away due to something called the seasoning sickness, which is basically, just like malaria or other endemic diseases that are living here, that are here. In addition, we have a very imbalanced sex ratio. So mostly male migrants were coming over, and this led to a lack of women in the colonies, right? So just think about this. We have a lot of people dying. We don't have a lot of women here. So how is this population growing? Well, it has a very low natural birth rate. So the only reason why the colonial population was increasing was because we just have a steady flow of migrants coming from England, right? So we have very low natural birth weight, but just a lot of migrants coming. So this can lead to a conclusion that, well, is kinship, biological kinship, how we relate to each other biologically, not a really important factor, right? Because everybody's passing away, we have a lot of populations coming over. We might get more of a mosaic or kind of a settlement history that's very different, a lot of unrelated people at the same site. However, though, when we look more closely at specific histories of specific counties, we can see that families are migrating within the colonial settlement. So, for instance, in Rappahannock County, Virginia, which was a relatively new place to settle in the 1700s, you can see it's actually very much predominated by folks coming, and as families, coming from different parts of the colonies. Right, so this is another lens where you think, okay, well, did biological kinship really affect the demographic history for these European migrants? Now, in addition, and a really important part of the story is the African diaspora, right? So for most of the 17th century, we have most of labor and being supplied by English indentured servants. However, in around 1680s, we get a really great economic spike in England, and nobody wants to be an indentured servant anymore, and we have a big drop in the number of folks that are coming over. And at the same time, we also get a rise in the importation of enslaved African individuals coming to the Chesapeake, okay? So this is a larger map of the transatlantic slave trade, of which is estimated almost 15 million persons were abducted and transported across the Atlantic. However, the Chesapeake is really only one very small sliver in this, right? So it's circled up on the top corner. Approximately 130,000 individuals were brought to the Chesapeake. So it's a very small amount, but also a very critical impact. Historian Lorena Walsh has gone through and looked at the registers of these slave ships to try to understand the regional origins of these individuals. And she can see that you can kind of partition that in Maryland and Virginia together, less than 13,000 individuals of African descent are living in the Chesapeake colonies by 1700. However, when you look a little bit more closely, we can see Maryland, most individuals are being brought from the upper West African coast, and those in Virginia are coming more from the central South coast, maybe Angola area, right? And now in Delaware, which is, again, this really weird peripheral state, we don't really know what's going on in Delaware. There's no major ports. Historians estimate that there are less than 500 individuals of African descent in Delaware by 1700. And we really have no idea where they're being brought from, likely being transported into a major port in Maryland or New York and then transported via land. So we also want to understand, what is the demographic history of African and African descended persons in the Chesapeake? So with these questions in hand, where we understand with archival and oral histories and archaeology, the what are we studying, the ancestry and relatedness in 17th century Chesapeake region for both the 17th century and its impact on the present day, well, we can try to look and try to use science, try to use DNA to understand the window and to looking at ancestry and relatedness. So this is where the Chesapeake DNA Project comes in. And, so who am I to, I got a very kind introduction from Dr. Bowe, so maybe we don't need to get into it too much. But a lot of my PhD thesis work was applying ancient DNA and studies in population genetics for both 17th and 18th century archaeological sites. So you can see them labeled here. St. Mary's is an African-American basilica in Norfolk, Virginia. We're also starting work at Jamestown at the 1607 Burial Ground Project, which I'm doing for my postdoctoral research, as well as Avery's Rest, and we're going to talk a little bit more about that later on. So my research is really focused on how do we use DNA to understand 17th century history? How do we use DNA to understand the history of our ancestors? And how do we look at DNA today to help us think about our past? So this brings me all to St. Mary's, right? Back, in 2019 when we did our DNA study, back when I was a young, spry PhD student, trying to think about a project, I kept thinking again about these stories, about these older families that had been here for generations. So I set out to create this Chesapeake Ancestry Project. We started in 2016. We got ethics approval from the University of Pennsylvania. We did a consultation with existing board members at the St. Mary's Historical Society in 2017, and on forth to get funding. And eventually, we're able to actually go forward and collect DNA samples in 2019. And we'll talk about that a little bit in this presentation. And, here we are now in 2022, where I think we're getting pretty close to finishing this project up. So I always like to talk about the timelines of projects, because I think sometimes when we see research, we're like, oh, the research is done. But really, this stuff has been going on for years, and we've been thinking about how to go from start to finish, right? It's hard for an academic to finish a project, because it's always so interesting, and you just want to keep it going. So today what we're going to do is I'm going to talk to you about the two threads, the two ways that we use DNA to study history. We're going to talk about the St. Mary's DNA Project, the modern DNA, as I like to say. And then we're going to talk about the ancient DNA. So this is the DNA of these archaeological ancestors. And you'll see both of these provide different perspectives on what 17th century Chesapeake genetic diversity is and what history is. And then we'll finish with conclusions about, what have we learned and where are we going. So now for the fun stuff. We get a little bit of a biology lesson tonight, so I hope everybody has turned their brains on. I promise. Peter said, I saw your slide set, Raquel. Man, there are a lot of complicated graphs. You're going to have to take it down. And I was like, don't worry. This is my favorite part about this, because I just love it. It's so fun. Okay, so in order to understand DNA results, you really have to understand what DNA is and how we get it, to understand how we can think about DNA, to understand our ancestry. So our DNA, this is our 23 chromosomes, 3.2 billion base pairs, is found in the nucleuses of all of our cells. So this is a little picture of a cell, right? We got our nucleus in the middle. And our 23 chromosomes, this is what we call our nuclear DNA, our total ancestry. So I get my 23 chromosomes from my mom and my biological dad. And every generation they get mixed up about 50%, right? And then my parents got their DNA from their parents and so forth. So really when we think about nuclear DNA or autosomal DNA, we can think about our total parentage, our total ancestry, right? The DNA of all of our ancestors. Now, we also have something called a mitochondrial DNA, right? So not to throw you guys all for a loop, but we got two different types of DNA in our bodies, right? So the mitochondrial DNA is a very small plasmodial DNA. It's about 16,000 base pairs, so very, very small in comparison to our whole genome. And it's found in the mitochondria of our cells. So maybe everybody has some kids in school and they learn mitochondria is the powerhouse of the cell, right? Produces energy, right? Well, mitochondrial DNA is associated with energy production. Now, the interesting thing about mitochondrial DNA is that it is inherited only from the mother. And that's because it can't be passed down through the sperm. So we only got to stick to the ovum, okay? So this allows us to look at maternal history, the history of the mother. So my mitochondrial DNA is the same as my mother's and is the same as my grandmother's. So it provides a window into one line of my family tree, right? So in comparison, think about our nuclear DNA. It's total ancestry. Mitochondrial DNA is our mother's line, right? Now on the flip side, we also have a Y chromosome DNA. So all biological males in the room will have a Y chromosomal DNA, and they get that from their fathers and their fathers before them. So this provides a perspective on male history, on paternal history, okay? So we can use DNA inheritance patterns to look and get different views of our ancestry through time, our total ancestry, our mother's history, and our father's history. So as folks, as modern humans evolved in Africa, right, some folks left Africa and with them spread their genes, right? Spread their genes across the globe. Oh, and this is a little bit whited out, so sorry about that. But this is supposed to be a map of Africa, and there's the Middle East and going into Asia. And you see kind of these movements, these circles with all these colors and letters, and how we talk and categorize this DNA is something called haplogroups, right? So haplogroups is a way for us is a nomenclature system for us to say, this DNA is really similar to that DNA, and we should group them all together, right? So it's a way for us to think about similarity, right? So when we see people with, let's say, haplogroup Q, we have this letter Q, Well, these groups of folks went out of Africa and then maybe are settling, I think this is supposed to be a picture of Asia over there. So we would look and see, okay, well, this haplogroup, what's my haplogroup, and where is it found in the rest of the world, okay? Now, how we would use genetics to think about relatedness, right? So we're thinking about shared ancestry, where folks are moving, what similarities of DNA do we have? Well, we can also use that to look at relatedness, right? So thinking about my mitochondrial DNA is the same as my mother's, so we're going to have the same haplogroups, right? The same ones. So this is how we have genetic genealogy. Folks that will use genealogy to say, I have this line, and then trace and find somebody that shares the same mitochondrial DNA haplogroup, because you can trace the same lines, right? So I always like to talk about this because when we have similar or the same haplogroups, right? Let's say I have an H and you have an H. That could mean that we share the same grandmother 3 generations back, but it also could mean that me and 50,000 other people could also share that same grandmother 50 generations back, right? Because we're talking about the movement of thousands of people, right? So we have to be careful when we're thinking about relatedness using mitochondrial DNA, using Y chromosome, because even if it's the same exact match, it might not mean you have a direct genealogical match, okay? So DNA and genealogy are saying a little bit slightly different things. And that's because this map is showing us here, we have a lot of the same haplogroups spread across the globe. A lot of people share the same haplogroups of DNA. Okay, so we talked about how we think about ancestry and relatedness using mitochondrial DNA, right? Our uniparental, the mother's and the father's history. Now we're going to think about it with our total ancestry, our whole genome DNA, right? So this plot looks very complicated, but I promise it's not, okay? This is called a principal component analysis, or PCA for short. And what this basically does, it's a visualization of genetic variance, right? How similar our DNA is to each other. One dot represents one person, one color represents one population. So you can see we have all these little purple dots and they're circled together. That's our sub-Saharan African individuals. In the top corner, we have a bunch of green dots. Those are all Asian populations. And let's say in the red here are European populations. So you see the different colors are grouping or closer together. So the closer the dots are together, the more similar they are, right? So you can see here, we have geographical groupings, okay? So what we theoretically do is we run one of these visualizations and then we would plot, let's say, my DNA on this graph and see where it falls. So for me, it would likely fall with European populations because that's my history, right? But for somebody else, it might plot, let's say, with Asian populations. So this is how we would assess ancestry using our whole genome. In addition, we can look at relatedness. So if anybody is on Ancestry DNA, you might have seen this shared centimorgans project. Is that right? Yeah, right? So the shared centimorgans project is basically saying, well, how much DNA do I have in common with, let's say, somebody else who's uploaded their DNA sample? Because if we think about if me and another person share, let's say, 50% of our DNA together, well, then we're maybe our brother and sister, and we just didn't know about it. So the more DNA that we share in common with somebody, the more likely we are related to each other. In biology speak, we call this identity by descent, or IBD. Basically, thinking about, are we sharing the same DNA, and is it coming from a shared ancestor? Or is this something that just arose randomly? So just to keep this in mind, this is the types of things that we're going to be trying to do in the future. Okay, so that's kind of our theory for the day, so thank you for bearing with me. We wanted to think about ancestry and relatedness in the context of living populations in St. Mary's County and Maryland that all share the same genealogical history, right? Again, thinking about folks who have ties to the 17th century through family lines, through oral histories and stories. So what we did in 2019 was collect DNA samples from folks that know that they have genealogical ties to the 17th century. We did this in St. Mary's Historical Society in Tudor Hall, where we provided consent forms and we collected genealogy forms, and each individual was interviewed for about 5 to 10 minutes to hear about their family history and to see the documents where they can show this line. We collected this genealogy form also contained information about grandparents, grandfathers, and then their founding Chesapeake ancestor, so if it was a Fenwick or somebody else. And then that way, in the future, we'll be able to kind of compare and see how much overlap in these founding ancestors is there. So in total, we enrolled about 125 people, which is really great. We collected 2 buccal swabs, so 2 cheek swabs from each individual. One was sent to National Geographic to do the Genochip 2.0 genetic ancestry test. Now, unfortunately, this test has now been discontinued, which is a huge bummer, and we were kind of a race against time to get those results in. But basically, this is kind of a test like 23andMe or Ancestry DNA, right? And then we took the other DNA swab and took it to Penn to do mitochondrial DNA analysis. And this was really to just make sure that we were getting the right results from the Genochip test, kind of a check to make sure. So what did we find? Well, first, I'm going to show you guys a series of graphs that we're going to first talk about Y chromosome, and then we're going to talk about mitochondrial DNA. So our Y chromosome, again, is our male history. And what do we see? We see a lot of folks with this R1A1 or R1B haplogroup. We had 59 men in the study. They're all kind of having the same haplogroup. And this haplogroup is found commonly in European populations. So the second map here is a heat map. You can see you have a lot in European populations, but we do have a locus in Africa as well as in the Near East, right? So this is saying that the majority of participants have European paternal history, okay? Now we're going to get to the mitochondrial DNA results, which just show an incredible range of diversity. I mean, completely unprecedented. Somebody who is coming into this as a researcher, I thought we were going to see kind of a lot of the same haplogroups, a lot of the same ones, because everybody has this shared genealogical history, these shared genealogical ties. Well, in fact, it was the complete opposite. So we see all these different letters are all different haplogroups. They're found all usually around Europe, but we also have African haplogroups. So we're going to basically break this down. So all of these different L types are haplogroups found in Africa. And you can see we have a lot of different types, okay? So these range from L0 all the way to L4, which is the entire range of African haplogroups that are currently found. They range from being present in Central Africa to Northern Africa to Eastern Africa. So we have a huge range of African diversity present, which is amazing. Now, haplogroup U, here's another example. Haplogroup U is commonly found in Europe, right? Very wide distribution. I mean, do you see how very many different types we have of U? I mean, it's incredible. And so, and there's only a couple of instances where we have 2 individuals that share the same haplogroup, right? So again, we have to go dig a little bit deeper to make sure to see if these folks are actually related or whether they just share the same haplogroup by chance. And that's where that genealogy is going to come into play. Now, H is one of my favorites because you look at this pie chart and you're like, how many different colors can Microsoft Excel use to make this pie chart? Well, not enough. We have a lot of repeat colors. So haplogroup H is a common haplogroup found in Europe. And it's a kind of a recent haplogroup. That's why there's so many different branches on it. And we can see in this graph here, we have so many different types. And this indicates that we have so many different lineages here. I mean, this is incredible. I just thought we were all going to get the same one. And then here we have this beautiful color wheel. So I think we have a lot more to learn about why we see this pattern here. And it's still a little bit confuses me. And so this is where I'm saying we're kind of in the middle of the research project because I get that result and I'm like, what? I thought, we're all going to get the same. So, we have these shared ties, this shared genealogy. Everybody has at least one ancestor to the 17th century Chesapeake, and yet our DNA results are telling us something completely different, that people are coming from all over. And so maybe the DNA from this modern study is maybe reflecting more recent migration. Or maybe the migrants in the 17th century were coming and had very, very diverse genetic diversity in the 17th century that we just don't know. So we have much more work to do. So we're going to have a student intern this summer, a master's student, and she's going to be digitizing the genealogy forms that we collected from study participants in order to try to make kind of a graph or connection chart to connect all the founding Chesapeake ancestors to see if we have nodes of overlap. In addition, we're also going to look at that autosomal data, that whole genome data, the total ancestry, to see if we see any relatedness between folks. Will we or will we not? I don't know. I think we might not, given how diverse this mitochondrial DNA results are. But this will help us think about how has genetic diversity changed since colonization in 400 years. Nobody has done a study that's local in the Chesapeake area in North America. So we really have no idea what genetic diversity looks like for Northern European American populations. Okay, so we're going to likely, I think, see a lot of diversity. So we'll be back. This is not the end of this story, and we're hoping that we'll get a lot of progress done this summer and this fall, and then be able to report some final results to you guys maybe early next year. So stay tuned. So now I'm going to share with you guys another way of thinking about the past. Modern DNA is throwing me a little bit for a loop because it's a little bit more complicated than I thought it was. But ancient DNA is a little bit complicated too, but provides a direct window into 17th century history. It's extracting and sequencing and analyzing DNA from 17th century archaeological burials, right? So we don't have that 400 years of additional genetic diversity that we see today, but rather coming right from the source. So I'm going to talk to you guys a little bit about my work at Avery's Rest. So this is up in the Delaware Peninsula. I guess it's kind of that part here. But again, this is part of a larger initiative to think and characterize the genetic diversity of other 17th century archaeological sites to understand and deepen where are people coming from? Are they related to each other on these archaeological sites? Sometimes when we have burials, we often don't have good documentation about who they are. So they're kind of just lost in history. So DNA provides a way for us to kind of reconstruct this past. Okay, so Avery's Rest is located outside of Rehoboth Beach, Delaware, if anybody has been. Really nice place to go for vacation. Also really great for DNA preservation because we got some sandy soils and it means really good DNA preservation because it's nice and dry. So Avery's Rest is a 1670s to about 1715 site in Delaware. So that's pretty early for British occupation. The site was first identified in 1976 through some site scatter. But excavations of the site did not begin until 2006 by Dan Griffith from the Archaeological Society of Delaware. So you can see they're setting up for their field season. So multiple field seasons have occurred and have identified many different archaeological findings. This includes structures, such as the red kind of boxes, the solid lines. We have at least 2 structures present at this site. The red circles are refuse pits, so things like trash pits, if people are throwing food away, as well as cellars where they're storing farming tools and other food sources, and at least 2 fenced compounds, so likely maybe for animals. So this suggests that this was a domestic site, right? So this is being occupied by local people, and they were living there. Now, importantly, 11 burials were also identified. So you can see them here in blue. And they were separated into 2 clusters. We have a group of 8 in the south and a group of 3 in the north. And they're really only separated by 15 to 20 feet. But you can clearly see they're being interred in the same space, but slightly separate from each other. So I was brought in, I think about 2016, to try to do some ancient DNA work on these individuals, because DNA can provide information on ancestry and relatedness, right? It can also help us think about genetic sex, right? So if you have a Y chromosome, you're most likely a biological male. So we wanted to understand what are the ancestries of those individuals that are interred at Avery's Rest, and were there any are they related to each other between these burials? Now you can see me suited up here in, very fashionable attire. This is, we have to be really careful about contamination in ancient DNA. We'll talk about that in just a second. We're all used now to wearing masks because of COVID. Well, in an ancient DNA lab, we got to wear masks. We got to wear a full suit. We got to wear double gloves. We can't have anything that's exposed during the process to make sure that we're only extracting the DNA of the archaeological individual and not like myself. Right? So where can we get ancient DNA from? Well, ancient DNA is really anything that's 50 years old that's been in the ground. You can get it from human skeletal remains, from bone. So teeth are a really great source, the tooth roots, it's very dense bone, as well as something called the petrous bone. This is a bone in your it basically protects all of your inner ear bones. It's in the temporal bone right here. And it's really, really dense bone, which is great. So the denser the bone, the better the DNA preservation. But we can also get DNA from things like coprolites or fossilized poop. We can get it from hair. We can get it from the inside of ceramics. Any organic compound that survives and preserves, we can potentially get DNA from. However, the DNA that we do get from these archaeological remains is often very fragmented and found in very low quantities, with a lot of other things that we don't necessarily want with it. So for instance, this is an infographic of what we see typically in an ancient DNA sample. In gold is the ancient DNA. In black is the microbial DNA. So things that in the soil, soil DNA, fungus, all that stuff also can get extracted with us. And we also sometimes can get modern human DNA. So that's in light blue here. So all of these other different kinds of DNA can really overwhelm our ancient DNA signal. So we have to be very careful when we're doing this extraction process not to make it any worse, right? So all this analysis has to take place in a designated ancient DNA lab. So this is one in the University of Tennessee. So an ancient DNA lab is a DNA-free lab, as much as we possibly can. It's cleaned with bleach. We love bleach in ancient DNA land. Kill everything, right? Because we don't want any living DNA or anything living coming near our sample. We'll fully clean the full room. We also have overhead UV lights, right? UV will act to bind together any modern DNA, so it can't be amplified, right? As well as everybody who comes into the lab has to wear the full suit, right? So it's kind of like being in a space lab or something like that. Okay, so that's how we basically go through how we get DNA, right? So what did we find here? Well, first we looked at genetic sex, right? So we were able to fully and successfully extract the DNA from all these 11 burials with good amounts of DNA, and it looked exactly like it should, which is great. So first we looked at genetic sex, and we were able to identify 2 females, both found in the southern burial cluster, as well as the infant and child. So there's a child in the northern burial cluster, about 5.5 to 6.5 years old, was assessed as a male, as well as an infant that was also assessed as a male, right? And that's because we can look at osteology of the bones to help thinking about sex, right? But when we have juveniles or people that are not fully sexually developed, it can be much more difficult to assess sex. So that's why DNA is really great. So here we have mostly men at the site, and we have 2 women, okay? So next we thought to look at ancestry, right? So where these people are from. Here's another PCA graph, right? So again, remembering 1 dot represents 1 individual, colors represent populations. And the black dots represent the ancient DNA individuals from Avery's Rest. So we can see we have 3 individuals that are up in the top right-hand corner, clustering with the orange dots. Those are African populations. And we have a cluster of 8 individuals at the very bottom of the plot with the yellow dots, and those are European populations. So this suggests we have European individuals here and African individuals here. which is very significant because if these individuals are of African descent, these would be the earliest recovered African or African descended individuals in Delaware. So we wanted to dive a little deeper with the European individuals. Well, if we know that they're likely of European descent, well, maybe what population in Europe are they coming from? So again, here's another great PCA plot. Again, each dot, one individual, same color, same population. And we can see the black dots here are clustering with these pink and this kind of teal blue. And that's populations in the UK and also in France. So this indicates that these individuals are of Northwestern European ancestry, right? So when we look at the archival documents, we know that Delaware was being settled by British persons, right? So this would generally fit with that interpretation. Now, you may ask the question, well, why French populations, right? Well, that's because Europeans are a very homogeneous population. We are moving around a lot and mixing our genes a lot. So we don't actually have a ton of genetic diversity because it's kind of shared with everybody else. So it makes it a little bit harder to get at that specific locus, that specific country, right? So that's probably why we're seeing both English and French, because we're probably moving around between the two countries, okay? So now we're gonna look at the African individuals, another PCA plot, right? I've starred these so you can see a little bit clearer where they fall. But one individual, AR10, very closely clusters with West Central African populations. So these are populations in the Gabon and Angola area, right? And then we see 9 and 11 that are kind of clustering a little bit in the middle, but mostly with West African populations. So this indicates that we have individuals of different African ancestries being interred here. They're not all being brought or coming from the same area. They're being brought from different areas within Africa, okay? So that's our autosomal ancestry, right? So we know we have European individuals being buried here and individuals of African descent being buried here. Now we're going to think about relatedness, okay? So here's a bunch of statistics about each of the IDs is one of the burials, what burial group that they're in. Coverage basically means how many times we have each position in the DNA covered, right? So greater the number is better, right? So we have some pretty high numbers here, which is great. And then we are going to look at our haplogroups. So for the first 8 burials that are found here in the southern burial cluster, we have a lot of letters and numbers that are associated with European populations, which fits with our autosomal findings. Now, interestingly, 3 individuals share the same mitochondrial DNA haplogroup, okay? 2, 6, and 7, okay? So that may suggest relatedness, but we're not sure. We're going to be skeptical about it. Now, 3 individuals in the northern burial cluster have African mitochondrial DNA haplogroups. Fits with our autosomal findings, right? However, none of them are the same, so they cannot be directly maternally related. Now we're going to look at our Y chromosome haplogroups, right? We see we have a lot of R1b1s, we have some I groups. These are also associated with European populations, but you notice none of them are the same, so they can't be paternally related. Okay, or direct paternal relatedness. Now let's look at the African individuals. We have African Y chromosome haplogroups, so that means African paternal history, and 2 individuals shared the same Y chromosome haplogroup. It's ding, ding, ding. We may have some relatedness here. So let's look at the autosomal results. And this will be able to more directly assess relatedness. So we have this is basically a graph that is going to show us the degree of relatedness between individuals. So if we have a dot that falls between first degree and identical twins, it'll mean it's a first degree relatedness. So that could be parent offspring, or it could be sibling, right? Anybody that shares first degree relatedness. Or second degree, that can be like grandparent, grandchild, okay? So basically what this program is doing is comparing how similar each of these DNAs between each of these archaeological individuals are, right? So we can see 10 and 11, their dot falls between a first degree relationship, okay? So this can either be a parent offspring or a sibling. However, we know that they share the same Y chromosome haplogroup. So this indicates that we have a father and a son being buried here. 11 is this 5.5 to 6-year-old child. So we have a father and a son, which is the first evidence of biological kinship using ancient DNA in North America. So it's hugely significant to think about relatedness, to think about what relationships were like, especially for individuals that were likely of enslaved status. Now let's look at our European individuals. So again, here's another plot that's showing relatedness. Individuals that are highlighted in red, I know it's a little bit hard to see, are first degree relatedness, parent offspring, and those in green are second degree relatedness. So this could be grandparent, grandchild. So we have 3 different pairs of individuals, and when we line them all up, we see we have a grandmother, mother, and a child buried here, because all of these individuals shared the same mitochondrial DNA haplogroup, that H1A1AF, right? And so when we layer these all together, we know that AR6 is about a 45-year-old woman. Her bones were crazy. She had like 3 teeth left. Her bones were really strong. And we have a 35-year-old mother, and then we have that infant, right? So here we have evidence of a 3-generational relationship between the European individuals. So this really helps us think about site interpretation, colonial frontier organization, with the 3-generational parent-offspring relationship for the European individuals, indicating that we do have evidence of biological kinship at these archaeological sites. However, we have a lot of unrelated males that are buried with them too, right? Isn't that kind of funny that we have only 2 women are related to each other at the site, but all the men that were of adult age are not related to these individuals at all. So this might suggest that we may have a loose biological kinship structure at archaeological sites that is supplemented by male labor, right? Folks are coming in, dying, maybe not reproducing, or whoever they're reproducing with, their children are not being buried in the same place. So this helps us think about, well, what did European American life look like? Well, it's kind of a mosaic structure, right? A very hodgepodge of people coming together. Now, for the African individuals, this finding is so significant in terms of thinking about what were relationships like between individuals. We are usually in any other ancient DNA study of folks of African descent, we typically do not see any biological relatedness. In another site I'm working on in 18th century Charleston, we see no relatedness between 36 individuals, and 12 of them are juveniles. So to see parent-offspring relationships is quite huge. And to think about their position together, right? This is 1680s. We have European and African individuals being interred together, whereas when we get to antebellum period, usually African individuals are being interred in a completely separate location, right? So this also helps us think about, well, what were relationships like in this frontier area between European and African descended persons, okay? So this helps us really zoom in into thinking about history, right? To thinking about what sites were like, what people were like back then. So, we have been able, what have we learned? So takeaway from kind of this perspective is that, this is our first assessment of early 17th century genetic diversity, right? And we know that folks are really diverse here, right? We got a lot of different haplogroups. And when we look at our ancient DNA analysis, we're actually able to zoom in on that 17th century story specifically to reconstruct ancestry and relatedness. So where are we now? We're kind of in the middle. I like to think, this is an ever-evolving research project. We're going to be analyzing other archaeological sites, such as Jamestown, to do these same types of analyses, understand where people are coming from and if they're related to each other, and then thinking about how that intersects with archaeological and documentary information. And with the modern DNA study, we're going to try to figure out what is going on and why we're seeing it, and report to you guys too, hopefully by the end of next year. So I just want to say thank you all for being such a great audience, for having me here today, for Peter for inviting me, and for all the funders that have been able to make this work possible. So I'd be happy to answer any questions. Thank you.
All right, speak up loud.
I was curious, and I'm not going to state this very eloquently because I'm not versed in all this, but when you compare the very colorful chart that you got with the local DNA, for instance, my husband, I
exactly right.
Repeat the question.
Yeah, so the question was, when we look at the local population diversity living today, we get that great, very colorful pie chart of all the different diversity. And then when we look at the 17th century, it looks a little bit different, right? And she was saying, is that because we have had multiple generations of people in between, right? In between the 17th century and to now, right? About 400, 300 years. And that's exactly right. I think that's exactly what we're seeing. And especially with the mitochondrial DNA, right? That's one lineage out of our total ancestry, right? It's one perspective going back. And that one line may not reflect that 17th century node that we're looking at. So that's why I'm really interested about the autosomal, about the whole genome DNA, because we're not limited to that one line, but we're gonna have to contend with all the hundreds of years of other genetic diversity that's come to play. But, what's interesting about that, is because it helps us think about, well, how has genetic diversity changed in 400 years? Researchers have been sampling contemporary populations across the globe to infer really ancient and old migrations. And I'm like, well, maybe that's not a very good proxy because folks have been moving, right? Especially here when even though we have a lot of deeply entrenched families and deeply entrenched histories, we're also a modern society, right? We all get on planes and move. So it may put a little bit of a hindrance in a lot of genetic studies, but I think that's a really important point to think about.
I have a question for you,
Yeah.
and an observation. But perhaps you can explain, when you were talking about the study that you did here in St. Mary's County, you spoke to the diversity of the DNA that you found. And it caused me to ask, is that diversity that you found, could it have been owed to a lack of screening of the people whom you tested in terms of their ability to know their ancestry back to the 17th century, which is what you were looking for? Or does it speak to something more fundamental?
That's another great question. And the reason why we did about 10 minutes of interviewing each participant is we wanted to control just for that factor, because we wanted to see to make sure we all know that there can be different levels and different rigors of genealogy, right? We can go on Ancestry DNA and just plug in families, and that's a perfectly good way of knowing. But we really wanted to make sure that the folks that were involved in this study really actually had documented lines to the 17th century. So when folks came and enrolled in the study, they brought documents with them, long family trees, and we talked through all of these documents, and they shared with me the stories that made their family. So the reason why I can pretty confidently say that this is not because of just maybe wrong participants enrolling in the study is because we really made sure to control for that. So what I think we're actually seeing is something a little bit different, right? I think this is the influence of maybe 200 to 300 years of diversity in all of our families that come in. Even if we're thinking about maybe one or two lines that trace back, we have hundreds of other lines of families that come from all over. So it's a kind of an interesting mental exercise, too, about what lines are we thinking about more often, and what are the other lines that are really making our DNA.
Yeah, I was thinking one of the issues would be tracing my family. It's through my mother's family, so it goes back to mom and grandmother and all that, but then ultimately it goes back to the male lines. That's who we keep track of in genealogy. So that's not going to match the mitochondrial. So maybe more work needs to be done we trace our family histories more through our mother's families and, forget all those guys, But I think that's where maybe some of that disparity could be coming from.
Right, because different lines look at different things. Our Y chromosome is going to look at our male history. Our mitochondrial is going to look at our female history, our mother's history, right? And they can tell completely different stories, right? Like we saw on those charts, the Y chromosomes all kind of looked a little bit the same, right? But the mitochondrial DNA was phenomenally more diverse. So that also might be a clue. So you guys are on the journey with me to try to figure it out, because, man, I was putting together this presentation. I just was like thinking back in my early journey, I was like, I know exactly what we're going to see. We're going to see everybody has the same. It's going to be boom, easy. And then to get these results, I was like, wow, I just was not prepared. So we're on a journey.
Question about, could you come forward a little bit? We want to be sure we capture your question. So if you would indulge me, just come a little bit forward, and I'll come halfway, and we can meet in the middle, I think. Yeah, don't pull it out. Oh my gosh, I gotta lose weight. There you go, thank you.
Hi, Mary Brennan. A couple of questions. Well, one question in particular. When you finish your research, will you go into then looking at the historical factors that might help explain why you got the results that you did? For example, in early Maryland, a lot of women died in childbirth, and many men had, several wives over a period of time. And then there was also the back and forth history of Maryland when the Catholics were exiled and the Puritans were brought in, and on and on and on. And you just had a lot of population swings over a period of time. So will you finish up with the historical context?
Yes. And that's why, we think back to the beginning of that presentation, we put the archival sources with the archaeology, with the science, right? Because I think the only really way to interpret these DNA findings is to look back at the historical record, right? The 17th century is an immensely complicated place. It's going back and forth, like, who is here? Is it a Catholic migration? Is it not? Is it tobacco? There's a lot going on. And we can look at different sites and different neighborhoods like Rappahannock County versus St. Mary's. They all have vastly different histories, right, of settlement. So I think we're going to have to look back at that. So I think you're totally right. But I also think we're going to have to zoom out a little bit too and think about, the larger demographics of the Chesapeake at this time being that high population turnover, high death rate, the lack of women, and how that also might give us some clues. But you're right on the right track. That's exactly how we're going to crack this egg.
So I have one, did somebody want to ask a question? Yeah, sure. Silas, here you go.
Excellent presentation. A question about the Avery's Rest population. 1680s, any evidence of the pre-English settlement over there, the Dutch and the Swedes?
Yes, another great. You guys are such a great audience. You're so knowledgeable at the time. So maybe a couple years ago, 2016, the archaeologist Dan Griffith discovered a pipe with the etchings of EB, which stands for, I think, Edwin Bird, right, which was a known Dutch tobacco pipe maker. Oh, yeah. So this really threw everybody for a loop. And we thought, and the reason why I was also brought on this project was because they wanted to know if this was a Dutch occupation or if this was an English occupation, which they thought they had documents on that showed who owned the land. But there's a lot of history of reoccupation of sites, right? So also on this land, we also see evidence of indigenous artifacts, right, of arrow points and pottery shards. So potentially this was also an indigenous site before it was occupied, potentially by the Dutch. We don't know for sure because it could either be a Dutch occupation and they were subsequently, the Dutch were in the Delaware and they were setting up trading posts. And then Maryland did not like that. And they came through and did a big raiding party and tried to kick them all out. And they burned down a lot of the settlements that were nearby. Again, not a great place to be in the 17th century. So they wanted to know, well, is this originally a Dutch site or is this an English site? So the pipe indicated, people thought it was maybe Dutch. But there's also a possibility of, trade between the Dutch are still in the Delaware, right? They didn't get wiped out. They're just in different areas, right? So it could just be evidence of trade. Who knows? This is what I'm learning from archaeologists. They find one artifact and they're like, oh no, it's a completely different site interpretation. But we have a lot of different artifacts. So still to be determined, but because we know from the DNA that these are Northwestern European individuals, likely they are of English descent. A great question.
So you sneaked two very subtle, made two very subtle comments during your presentation about Avery's Rest. I don't remember literally, but you, a couple of times, Raquel said, this was the first incident or the first discovery of. And so it occurs to me that you are the one who discovered those things. And it occurs to me that as a result of that, we in this room are looking probably at one of the giants in anthropology. That you found these first. No, I say that because that's pretty impressive. How many people in your field uncover the first of something?
Not very many, but it's still, so you also guys got a special preview because this stuff is not published yet, but we are submitting it to, I think, either Science or Nature soon, so fingers crossed. Yeah, doing work in this field in this time period, I think, is so critical to be able to think more specifically about the sites that we go to as children. Like for me, going to Jamestown or going to Williamsburg, you learn about the history of these sites, and now we have a different way of thinking about that history using DNA. And so it's going to offer different ways of thinking. Maybe it doesn't have to be the first, but I think it's important because it offers us a different perspective into the past, especially when we don't have good documentary records, especially for the histories of African descended persons in this nation, where we don't have a lot of information. This provides one way of kind of trying to step over that documentary barrier where we don't have it. Now, it's really important to do this work with communities and in consultation, so we're not just taking bones from folks and doing the DNA, but doing it in concert with folks that connect to these remains. So there's a whole other ethical debate that we did not even talk about today, but yeah, I'll just leave it at that.
So being a
Don't get wrangled.
Busby Dick is going to pull me over. All right, there we go.
okay, being a product of the '60s, if you will, West Side Story said, "Stick to your own kind, marry your own kind, stay with your own culture." Now that kind of disappeared between then and now, but then it was a big deal. Was it a big deal prior to that, where people did not intermarry between ethnicity and religion and so forth?
Well, it's hard to tell.
Yeah.
So, when we, I think about Avery's Rest, for instance. So we know that the European folks that lived at Avery's Rest, John Avery was born in England. He married Sarah Brown. She was born in Massachusetts, came down to, St. Mary's County, actually, St. Mary's City. And that's how they met, and then they got married. So they're, coming from very different areas, right? So they're not the come here folks, so you haven't been living in St. Mary's, we can't be married, right? There's a lot of mixing. Now, in terms of between African and European individuals or African and indigenous individuals, we can look at things like through the documentary record, but sometimes those records are going to be sparse. Sometimes the genealogy is not always right. We have a lot of issues with false parentage. So DNA may be a way to think about this. So I think about the 18th century African descended site I'm working on in Charleston. Most of these individuals are coming from different areas in Africa. We did the DNA on 36 of them. We have a whole group of individuals coming from the West Central African region, and then another group of individuals coming from the Upper Gold Coast. One individual has a Native American mitochondrial DNA haplogroup, right? And so we were a little confused by this. And then when we look at the autosomal DNA, the whole genome DNA, 30% of her ancestry is also indigenous North American. So if we date, because we can do this now, we can date when that admixture event occurred, and it happened in the 1680s between 1710. Between that time. So we have, using the DNA, a documented occurrence of interbreeding between African and indigenous individuals. Now, what the circumstance behind that is not going to be known because we don't have any records about who these individuals are, but we do see it in the DNA, but very, very limitedly. So, again, this is a story that, we're just going to, the more sites that we do, the more histories that we're going to uncover. I think it's kind of, people all get together with all different kinds of people, but we'll just have to see.
Any other questions for Raquel? Thank you, Raquel. You are a genius.
Well, thank you all for attending tonight. Just put on your calendars, October the 13th will be our fall dinner. We'll do this again. Thank you all. Be careful going home. Watch careful for the traffic. Thank you again for coming. Take care.